Is it okay if we start the interview right?
Sure.
You study mainly like lentiviruses, right? Like HIV and stuff.
Yep.
And lentiviruses and like coronaviruses are different kingdoms. So can you like compare them and see like what are some of the similarities, what are some of the differences, etc.?
hmm Sure. Similarities, right? When you think about viruses, we call them obligate parasites for a reason, right? They basically require a host in order to facilitate their life cycle. So something similar between coronaviruses and lentiviruses on the big picture is that they utilize a lot of host cellular pathways to facilitate their viral replication cycle, to make more of themselves, and to be able to spread. So viruses are really, really well adapted, well evolved to be able to steal the host machinery they need in order to facilitate their life cycle. Um, they've also really evolved how to overcome our antiviral mechanisms. Right, so we have within every single cell, there's an intrinsic antiviral pathway that we call the innate immune system, or the innate immune response to a virus. And we actually do a really good job. We have a lot of really good mechanisms to block viral replication, but of course, viruses are just better. And so they both coronaviruses and lentiviruses have evolved to overcome those pathways in really, really sophisticated manners. So that's kind of some of the ways in the big picture that we think about them as being similar. Um, there's a lot of differences in the specifics of the viral replication. Um, Lentiviruses are single-stranded RNA genomes, but they have this really kind of two really interesting aspects to their life cycle. One is that they take their RNA and they turn it into double-stranded DNA,
Oh,
which
interesting.
is very, very different. And this is a process called reverse transcription. And this really goes against the central dogma of biology, right, where you
Yeah.
go from DNA to RNA to protein. And lentiviruses or retroviruses, which is the family that they're part of, were kind of the first example that you could really challenge this really central idea of biology where you could actually go from RNA back to DNA, which was pretty cool.
Yeah.
And so the reason they do that is they then take their double-stranded genome, so they go from a single-stranded piece of RNA through this reverse transcription process to make a double-stranded piece of DNA. They import that double-strand DNA into the nucleus, and then they actually integrate that DNA into the genome. And so what that means is that when somebody is infected with a retrovirus or a lentivirus, that DNA becomes a part of them for life, or for the life of that cell. Because now they've taken their DNA and they've cut open your DNA, they've pasted their DNA into your DNA, and now their DNA looks exactly like your DNA. And so for as long as that cell is there, that cell will be having the genome of the lentivirus and will be able to make new viral particles. And so that's why things like HIV infection are lifelong, whereas something like a coronavirus infection, right? Virus is going to make more of itself. The cell is going to die, and once you clear that virus, the virus is pretty much gone, right? And so that's one really, really big difference in these viruses.
So just this coronavirus is just like standard normal virus that just attacks you and
As you know, we're learning a lot about this SARS-CoV-2 now, and it's actually a really fascinating virus. It's got a lot of tricks up its sleeve. It's got a lot of reasons why it's different than other kind of viruses. But in terms of the um, kind of time course of infection, I guess, right? So we think of viral infections on a few different ways. There are acute viral infections where the virus gets in, it infects, it makes more of itself, and then it kind of gets cleared and goes away, right?
yeah.
And then we have some where they'll infect and they'll slowly start making more of themselves over time. And then there are some that actually will infect, they'll go quiet, and then they'll be able to come back, right? So herpes virus is a case like that.
Yeah.
And so coronaviruses are, for the most part, in that first kind of acute type of infection where they come in, they make more of themselves, start to get some symptoms, your body recognizes that, starts to clear it, and then it goes away. But one of the things that's really fascinating about this current SARS-CoV-2 coronavirus is that, first of all, it seems to make more of itself, a lot more of itself, before the symptoms even start to come on, right? And so that's one of the big problems is that most people are making a lot of virus and shedding a lot of virus before they are showing any symptoms of being infected. Um, and the other aspect of that, what it does is it actually means that there's a delayed innate immune response to that virus, which has actually been what causes a lot of the pathogenicity of the virus. So, in one hand, yes, it's kind of this kind of quote unquote standard kind of acute viral infection, like you think of for, a common cold or flu or something like that, in that it is not a lifelong infection. But coronavirus definitely has these kind of unique aspects to its replication.
So, are there any signs, for example, like can you look at HIV and say this is a lifelong virus, or can you look at herpes and say this is a virus that pops up once in a while just by looking at like its proteins or its genome or anything like that? Is that
Yeah,
possible?
so there are certain uh, genes in the virus that facilitate those different steps. And so, for example, um, lentiviruses like HIV, they encode a protein called integrase. And what integrase does is it integrates its DNA into the host genome. And so if you were to take a virus and you were to say, oh, you know what, this virus has an integrase-like protein, you can start to guess maybe this is going to be a virus that is going to integrate and become lifelong in that regard. Um, Herpes viruses, they have a mechanism to promote something called latency, which is where the virus will actually still be there, but won't be making any more of itself, at least not enough of itself for our body to recognize that it's there. And so herpes viruses have an active process to push the virus into latency, so that the virus can go quiet and the body doesn't think the virus is there. And then they have an active process to push the virus back out of latency to make more virus at a later time point. And so if you could look again in the viral genome for the viral genes that are important for that, you could say, okay, this is going to be a virus that is going to have that kind of latent phase. And so for coronaviruses, we don't see any of those, and we know a number of coronaviruses, and we've never seen any of them having this kind of latent or lifelong infection.
That's really interesting.
Yeah, viruses are viruses are really interesting to me, and I've they are, I study HIV, right, which is
Yeah.
a huge problem globally, right, and it has devastated so many lives. And I understand the gravity of HIV infection, of SARS-CoV-2 infection, obviously this current coronavirus infection. There's so much gravity and so much heartache, and it's so important that we work to try and fight these viruses. But on another level, they're just so interesting. And what's really important is that a lot of the molecular biology that we know, we have learned by studying viruses, because viruses have figured out how our systems work, and they can be tools to teach us more about ourselves. So,
So, in your study in HIV, in your research, did you did you mainly look at, for example, like I think one of them you studied the protein in HIV. It's I forgot exactly what it was. Is I think it was like to inhibit one of our antiviral proteins.
mm-hmm.
And and did you mainly look at, for example, like did you infect cells with HIV? I saw you infect some cells with that. Do you mainly look at that, or do you also look at the genome and stuff like that?
Yeah, so we do. One thing I love about virology is we do a lot of different kinds of techniques. And so in my lab, we can work with full length replication competent HIV. And a lot of people ask me how we do that. And we can actually make HIV.
Wow.
And so we have different aspects so we know the genome of the virus, right? And we know what the virus needs to make more of itself. And so scientists before me basically took parts of the virus and put them onto what we call plasmids, so pieces of circular DNA. And then we can just put those plasmids, we can deliver them into a cell, and then the cell will use its normal machinery to make these proteins, and those proteins will make the virus. And so then we can collect that virus and we can use it to do different things. We can also take specific viral genes that we're interested in. And so there's two viral genes that we study a lot, VPR and VX, which one of them we have an idea what it does, the other one we don't know what it does. But we can take those genes in isolation, right? We can clone them, and then we can express them in a cell and try and understand a little bit better what it is those specific genes do. We can also, back to the idea of the full length virus, this replication competent virus, we can also now remove those genes from it and ask, how does that alter the viral life cycle, right? So we can do a lot of manipulation of the viral genome and viral proteins in order to really try and understand what it is these viral proteins do and how they facilitate viral replication. The other thing we do a lot of in my lab is that we're very interested in how these viruses have evolved. And not only that, but how they've actually influenced our evolution. Because while we may think of HIV as a pretty new virus, lentiviruses, that family, are millions of years old.
Oh, wow.
And they've been infecting primates for millions of years. So they've been infecting our ancestors and closely related species for millions of years. And so because of that, they've really shaped our immune system because we've had to be fighting them constantly. And so we really, our body has evolved to try and deal with these viruses for millions of years of evolution. And so we're really interested in kind of those two facets, right? How does the virus shape us and how do we shape the virus? And so we do a lot of kind of looking at viral sequences, looking at viral DNA, not only from humans, from HIV in humans, but also the family of the viruses that include these simian immunodeficiency viruses, or SIV, that infect different primates.
This stuff is like really fascinating. I can see why you went to virology.
It's fun, yeah. It really, it's yeah.
So, why should why should people care, for example, about sequencing the genome of any virus, HIV or SARS-CoV-2? Like, why does the average layperson why should they care about it?
Yeah, that's a really great question. So knowing the genome of the virus tells us a lot of things. And so, for example, SARS-CoV-2, I'll start off by saying research is being done on SARS-CoV-2 at a faster rate than I have ever seen in my life. People are doing science so fast right now. It is quite amazing and quite impressive.
Even faster than HIV or
Yeah. And so, for things that took, 10, 20 years for studying HIV, people have been doing in months for SARS-CoV-2. Part of it's because of the history, right? Because there's a few things. One is that we learned a lot by studying viruses like HIV, right? And so a lot of those tools and techniques that people developed and implemented in the study of HIV are now being implemented to the study of SARS-CoV-2. There's also the study of the previous coronavirus outbreaks, SARS and MERS, which has also allowed us to jump onto the ship a little bit quicker. But then there's also just the fact that it's a really, obviously, it's a hugely important virus right now at a very fast time. And so it really requires a very fast response. And so one of the first things that people did was sequence the virus. And the reason for that is to understand, what is this virus, right? And so when they got the sequence of the virus, what they could do is they could start looking at that sequence, and immediately, as you asked earlier, they could look at it and say, you know what? We know what this virus looks like. The genome of this virus is a coronavirus, right? Because we've sequenced other coronavirus genomes. We know the genes they encode. We know the organization of the genome. We can say, you know what? This looks very similar to other coronaviruses, which immediately tells us, okay, how are we going to go about dealing with this virus? How are we going to go about trying to fight this virus? The second thing that it allowed us to do is try to understand where the virus comes from, which is an ongoing question that I'm super fascinated by. Where do viruses emerge from? And so, originally we didn't really know what to call this virus, and we didn't really know where it came from. We still don't know where it came from. We know that it originally came from bats, but we don't know if there's an intermediate species that the virus jumped from bats into something else, into humans. But what we do know is that it's very closely related to the original SARS that emerged in 2003. And so that's why it's being called SARS-CoV-2 or SARS-2. And so by sequencing the genome, it also allows you to try and understand this puzzle piece of where the virus came from, right?
yeah.
Which is going to be really important in trying to understand how we can prevent this transmission in the future, right? By understanding where the virus came from, we can start to understand and potentially prevent it from jumping species, or a similar virus from jumping species in the future, right? So the third reason why it's really important to sequence the virus is trying to understand how the virus is actually spreading within humans and evolving within humans. So those two things we can really see by sequencing the virus kind of in real time, right? And so now not only are we sequencing the virus, but we know a date that that virus was sequenced, and we know a place where that virus was sequenced. So now we can start to understand not only the big picture of how did a virus jump from one species into humans, but now how is this virus spreading around, right? And we're seeing things like, for example, the majority of the virus in New York was coming from Europe, right? And that was because of a lot of this air traffic of people from Europe back to the United States, right? And we can see how the virus is potentially changing in people and what that means, right? So we can do real-time investigation as to how this virus is moving around the globe, which is really, really important, again, for thinking about how we can fight this virus, right? The fourth reason I'd say it's really important to sequence the virus is now we can actually study it, right? We can do what I love, which is molecular biology on the virus. We can make the virus, we can infect cells with the virus, we can isolate viral genes, and we can try and understand what they did, what they do. And so, for example, there was a recent publication from a group at UCSF, a colleague of mine who I actually just had a conversation with last night named Evan Krogan, who published a paper where basically they took every single gene, every single protein coding gene of SARS-CoV-2, they expressed it in a cell, they identified what proteins in the host that viral protein interacts with. So that's called, they're looking at the interactome of that viral protein. And then they identified what compounds, what drugs are already available that could potentially target that interaction between the virus and the host and potentially block viral infection, right? And so by sequencing the virus, we can now start to do some really important molecular biology to really understand not only how the virus works, but also how we can potentially target and treat the virus with drugs that are available. So,
It sounds like it's pretty important.
short answer, TLDR, yes.
I really appreciate you taking the time. It was really nice to meet you and stuff like that.
Yeah, you're very welcome. My pleasure.